Thanks, Anne. Welcome to the postprandial dip. I don't want to see any of you sleeping this afternoon. Yeah. Our first speaker this afternoon is somebody who you've already seen this morning. We're making her do double duty. Is Karen Lillycrop, who's professor of epigenetics in the Center for Biological Sciences at the University of Southampton in the UK. Dr. Lillycrop's research focuses on the effect of early life environment on the epigenome and the long-term consequences for disease susceptibility. She will discuss developmental plasticity, sensitive periods, and the risk for obesity.
Thank you. Yeah, so today I want to talk about the effect of early life nutrition on the epigenome, the periods of sensitivity, and then the implications of these altered processes and how they might impact on risk of obesity. Okay, so traditionally then it's been widely accepted, as we've heard, that our genes determine our phenotype and susceptibility to a range of chronic diseases in later life. However, rates of obesity, as you know, have risen dramatically over the last 20 years in both developed and developing countries and are projected to rise higher by 2030. Such a rapid rise in rates of obesity over such a short time frame cannot be explained solely by genetic factors, but it suggests that the environment also plays a role. And the mechanism by which the environment may influence disease risk, as we've heard, is through the altered epigenetic regulation of genes. As there is now increasing evidence that a number of environmental factors, including nutrition, can alter the epigenome, and the epigenome seems most susceptible to these environmental factors in early life, particularly during the prenatal, neonatal, and pubertal periods. And most of this evidence that early life nutrition can alter the epigenome has come from work from animal models. And these have shown that maternal dietary restriction, protein restriction, as well as maternal overnutrition can induce long-term metabolic changes within the offspring. And often what you see is both maternal undernutrition as well as maternal overnutrition induces very similar metabolic changes. So in both cases, what you tend to see is dyslipidemia, insulin resistance, and obesity in the offspring. And these phenotypic changes are accompanied by changes in the methylation of key metabolic genes. So for instance, if you feed rats a protein-restricted diet just during pregnancy, you see alterations in the methylation of a range of genes, including a gene called PPAR alpha. And this is the peroxisome proliferated activated receptor alpha gene. It encodes for a nuclear receptor, which plays a key role in lipid metabolism. And so what you see is a decrease in the methylation across the promoter region of this gene in the liver of the protein-restricted offspring compared to the controls. This decrease in methylation of this gene is accompanied by an increase in its expression and an increase in levels of beta oxidation, the metabolic process that it controls. And this change in methylation is seen as early as embryonic day 18 and seems to then persist right the way through into adulthood. But it's not just maternal undernutrition that can induce these epigenetic and phenotypic changes. We've also seen that maternal overnutrition can also induce such effects. So here, for example, we fed pregnant rats a diet either high in saturated fats or in fish oil. And we found this altered fatty acid composition within the liver of the offspring. Specifically, we saw a decrease in 22, 6N-3 and 20, 4N-6 levels in the liver of the offspring from the dams fed the diet high in saturated fats or in fish oil. As the FADS2 gene encodes the rate-limiting enzyme in polyunsaturated fatty acid synthesis, we then looked at its expression and methylation in the offspring. And we found that FADS2 expression was decreased in the offspring from the dams, fed the diet high in saturated fats and in fish oil. And this decrease in the expression of FADS2 was accompanied by an increase in the methylation of CpGs within the promoter region of this gene, including the CpG at -394, with methylation at this site accounting for over 56% of the variation in levels of FADS2 mRNA expression in the individual offspring. There is also, though, increasing evidence that this period of epigenetic plasticity extends into early postnatal life. So, for example, Andras Plageman showed that overnutrition in early life, which he induced by raising rat pups in small litters, induces rapid fat gain and obesity in the offspring. And this was accompanied by alterations in the methylation of CpGs within the promoter of the appetite control gene POMC. And specifically, what they saw here was hypermethylation of CpG sites, which lie within the binding sites of transcription factors such as SP1 and NF-kappa B. And these are important for the induction of POMC by leptin and insulin. And what you see in these offspring is, despite the fact that they have hyperleptinemia, there is no corresponding induction of POMC. So, many other studies have shown that variations in micronutrient intake, whether it's during pregnancy or during the peripubertal period, can also induce changes to the methylation of genes. And there have also been studies which have shown that variations in paternal diet can again induce altered methylation of a variety of genes. So such changes to the epigenetic control of these genes, which often play key roles in metabolism and appetite control, have suggested that maybe these changes do indeed underpin the long-term changes that we see in metabolism and disease risk in the offspring. But what then about us? Does the same phenomena occur in humans? Well, a number of groups have started to address this, and many groups have looked at the effect of the Dutch hunger winter, because back in the Netherlands in 1944 there was a famine when energy intakes dropped from 1,800 kilocalories per day to between 400 and 800. And what subsequent studies have shown is individuals born to mothers exposed to famine during pregnancy have an increased risk of developing heart disease, diabetes, and obesity in later life. And what they also found is alterations to the methylation of a number of genes in those individuals who were in utero exposed. Specifically, they saw an increase in the methylation of interleukin 10, GNAS, leptin, ABCA1, and MEG3, and a decrease in the methylation of IGF2. And interestingly, these measurements have been made 60 years after the famine, suggesting again that variations in maternal diet induces this very persistent epigenetic change within the offspring. This ability of early life nutrition to alter the epigenome has been suggested to be part of a normal adaptive process, a process called developmental plasticity, when an organism responds to the environment in early life, adjusts its developmental path to produce a phenotype that confers a survival or fitness advantage. So, for instance, poor maternal nutrition might signal to the fetus that nutrients are scarce, in which case the fetus will adapt its metabolism, it will reduce energy demands, it will increase its propensity to store fat, put less investment in bone and muscle mass, preparing, if you like, for an uncertain life course. If that organism now finds itself in an environment where nutrients are indeed scarce, its metabolisms will be matched. It will be of low disease risk. If, on the other hand, it finds itself in an environment where nutrients are now abundant, its metabolism will be mismatched. It'll have this increased propensity to store fat, be at increased risk of metabolic disease. And this mismatch between the pre- and the postnatal environment has been suggested to account for some of the rapid rises we've seen in rates of obesity and diabetes in the developing world as populations move from rural to urban areas. So there is growing evidence then from both animal and human studies that early life nutrition can alter the epigenome And these reset marks then appear to persist, contributing to alterations in metabolism and disease risk. So if this is the case, then it should be possible to detect these changes in early life and use these then to estimate metabolic capacity and disease risk. The only issue with doing this in humans is there is limited tissue availability. So readily available tissues that we could access would be obviously cord blood, placenta, buccal and blood. And there's obviously a real question mark whether the methylation in these cell types adequately reflects the methylation in more metabolically relevant cells. But maybe if the environmental constraint occurred early enough in development, then all three germ layers might be affected. So we decided to look to see if we could find methylation differences at birth that were associated with later adiposity. So we started off by sequencing the promoter regions of genes that are known to play a role in adipogenesis in cord tissue from 2 mother-offspring cohorts based in Southampton. And we found that the methylation of a CpG within the promoter of the retinoid X receptor at birth was associated with percent fat mass of children aged 9 in the Princess Anne cohort and age 6 in the Southampton Women's Survey. So this supports this idea that these developmentally induced epigenetic marks might indeed make a significant contribution to later phenotype. And it also suggests that it may be possible to detect such marks even in peripheral tissues. So given that the identification of such marks would be extremely useful to identify individuals at increased risk, we then carried out a genome-wide screen to look for methylation differences across the genome that were associated with percent fat mass of the children aged 6. And we used an MBD array approach, and we identified 93 differentially methylated regions, or DMRs, at birth that were associated with percent fat mass of these children aged 6. And we found that 41 of the 93 DMRs were associated with genes that play roles in DNA replication and repair. And one of these DMRs was located within a gene called CDKN2A. And this is a gene which is known to encode for 2 cell cycle inhibitors, and it's known to play roles in proliferation, differentiation, and senescence. So to validate this DMR, we then sequenced the CpGs within this region in cord tissue from a separate group of children from the same cohort. And we found that lower methylation of CpGs within this DMR at birth were associated with percent fat mass of the children aged 6, so consistent with the array. We found a similar association at 4 years. So then to replicate this, we then looked in an independent mother-offspring cohort, and this was the Gusto cohort from Singapore. And again, what we found was that methylation of CpGs within the DMR of CDK were associated with an increase ponderal index of the children at 18 months. So this again supports the idea that these developmentally induced epigenetic marks might be important contributors to later phenotype. But while such marks would be extremely useful, ideally what one would want is a mark that could be also used to follow the progression of the altered phenotype through the life course, and then one which could be used to monitor the effectiveness of interventions. Such a mark would have to be present in tissues that you could readily access through life, such as blood or buccal, and also it would have to be relatively stable. And although DNA methylation was originally thought to be a very stable mark, so once set up in early life was then thought to be stably maintained, there have been a number of recent studies which have suggested that maybe it is more dynamically regulated than previously thought. So studies have shown that just doing an intense bout of exercise or even just going to the city for a weekend could change the level of your methylation. So if this is the case, then maybe this might limit the use of an epigenetic mark to follow the progression of a phenotype through the life course. So we wanted to look at the question of stability, particularly during childhood, which might be a very plastic period. So we've started to look at the methylation of genes in blood samples taken annually from children aged 5 to 14. And the genes that we looked at to start with were Saturnin-1 or SIRT1, PGC1 alpha, and HNF4 alpha. And we chose these genes because they play a key role in energy sensing and then in the coordination of the metabolic response. And we found that the methylation of the CPGs in the promoter regions of these genes was stable over this time frame, suggesting that at least for these sites, methylation is set up in early life, in this case, before the age of 5, and then stably maintained there afterwards, despite the onset of puberty, variations in exercise or pollution. So having found these CPGs that were stable, we then looked to see if they were associated with the phenotype of the children. And we found no association between the methylation of CPGs within the promoter region of HNF4 and adiposity, but we did find a significant association between the methylation of 2 of the CPG sites within the promoter region of SIRT1 at 5 to 7 years, with percent fat mass of these children from 9 to 14. And we saw this effect in both boys and girls, despite the fact that in boys, fat mass declines, whereas in girls, it increases. We also found associations between the methylation of 4 of the CPG sites within the promoter of PGC1 alpha at 5 to 7 years, with percent fat mass of these children from 9 to 14. And again, we saw this effect in both boys and girls. And here, the magnitude of the effect was such that for a 10% difference in methylation at 5 to 7 years, percent body fat differed from between 6.3 to 12.5%. And interestingly, what we also found was an association between the same CPGs within PGC1 alpha and SIRT1 in cord tissue at birth with measures of adiposity in the SWS cohort as well. So we saw associations with skin fold thickness and also total and percent fat mass. And we saw these associations going from birth through to 6 years of age. And this was the oldest age that we had measurements of these children from. So this finding of an association in both cord and in blood does suggest that maybe there is an environmental imprint on the epigenome, which is then detectable across tissue types and time. But the question remains, so is this or are these alterations in methylation that we see, are they simply markers of adiposity, or do they actually play a role in the development of obesity? So to begin to address this, we've started to look at the effect of methylation of these CPGs on the function of PGC1 alpha. And we found that if you methylate the individual CPG sites, so the ones that were associated with adiposity, this leads to a decrease in the promoter activity of PGC. And what we also found was methylation of the CPG site at -783 altered the binding of the HOXP9 PBX heterodimer. And this is a complex which is known to be important for adipogenesis and is known to be an important regulator of PGC1 alpha, suggesting that maybe methylation at these sites might have a functional consequence. So then in summary, there is now growing evidence that early life environment is an important determinant of later obesity risk. And there seems to be 2 pathways. There's the undernutrition pathway and the overnutrition pathway. There is also evidence that early life environment can alter these epigenetic marks, and these altered marks appear to make an important contribution to later phenotype. And it may also be possible then to detect these altered marks in early life and use these to identify those individuals at increased risk. But there is much we have yet to learn. For instance, we know very little about the pathways that nutrition or over nutrition leads to obesity? What are those epigenetic changes that occur? Are the signatures the same in each pathway? And of course, it's likely to be not just a change in methylation, but also non-coding RNAs and also histones as well. And also, what are the drivers that induce these changes in methylation that we see? Nutrition is obviously very important, but what are the other factors as well? And of course, are there marks that we can change? And if so, when and how can we intervene? But I think we're now in a position to begin to address these issues, either with the cohorts that we have or by establishing new ones. And if we can begin to address some of these issues, we should then start to be able to stem the tide of this ever-increasing rise in rates of obesity. Thank you.